that we have. Next week, we're going to go over your story maps individually. So what I'll do is like right at the beginning of the class or like a couple minutes before class starts, I'll sign up like I'll send out like a Google sign up, like a spreadsheet or something. And you guys can sign up for times. Here's what's going on. Um, I owe you homework 5. I owe you map assignment 2. You all are about to owe me your story map drafts. And so let me look at the calendar, roll it quick. And I had that set at this Friday at noon. And the reason that I have your story map draft due Friday at noon instead of Tuesday at noon is because you all are going to have an assignment associated with lab today. All right. It's not going to be a map assignment, so your soul doesn't have to be crushed, Lauren. It's not going to be that bad, right? But you are going to have it'll be like a homework because I got to get 40 more points. Okay. That'll be something small. It'll be something, right? But does making the story map draft do like Sunday help anybody instead of Friday? I just need to be able to grade it on Monday so that I can be ready to look at it with y'all on Tuesday.
That works for me because I've got a test and a paper due on Friday.
Yeah.
So
It sounds pretty bad for everybody. It sounds pretty bad. Okay. Yeah, I'm so sorry. We do this everybody does all of this at the same time. Yeah, okay. Okay. Okay, so yeah, so you're gonna have your story map draft is gonna be due on Sunday. I'll change this right now. Sunday. That's probably the 24th. Um, and so the thing with your story map draft is you don't have to necessarily be in the computer lab to do it, right? Because this is all going to be done um through through your online ArcGIS stuff. Okay. So if you're actually creating any maps, what am I doing? Sunday 24th at midnight. Um, if you're creating any maps, remember the process of doing that is you have to make your shape files or work with your raster data in ArcPro, upload those files to your content, and then you build your interactive map in ArcGIS Online. Okay. So listen, I don't expect necessarily everybody to have their maps done. Um, Having an idea of what your map is supposed to be, including like figure captions, discussing where your data are, something like that, that's going to be super helpful, right? But think about the story map draft as just like as much structure of your story map as you can get there, and then try to make some maps, or at least make sure that you have your data all organized and you kind of know what to do. So because that's one of the things that I want to talk about next week in class is if you're like, I really, because so many of y'all, I gave you your feedback back for your story map outlines. You've got that feedback back. And if for some reason you didn't see your feedback, remember that you always have to turn on track changes and you see all markup in that Word document that I provided back. So you're going to want to look at that. But a lot of y'all, your ideas for maps were like, I'm going to recreate the range map. I'm going to recreate the distribution map for your species. And that is fine. However, right, we are a habitat focused program. You all increasingly will be in habitat focused positions. think about what your organism is. If you were a person that's like, I have a specific organism, what are the habitat conditions that either encourage or prevent the establishment and distribution of that organism? Because increasingly, what we're going to be left with is we're going to need to be doing mapping and visualization and assessment of habitat, right? Instead of just unique species. So really think about how and where you can get habitat information that is relevant to your conservation issue and or your species. Um, so just get that in so that I can, because if I can't grade it, then we're not going to have, it's just going to be like me looking at your story map while we review your, while looking at your story map draft while we review it like in real time, which I mean, it's not the worst thing in the world, but get it in so that I can look at it ahead of time. This is graded, it's 50 points. Um, and what you're going to do is you're just going to build your story map. You guys have had all of those exercises in story maps part 1. You've got all the details in your story map section here, right? You've got your planning workshop, you've got practice, you've got the draft, you've got your rubric that you can take a look. Do I have a rubric? There's something in here. Let's see. Did I provide it for y'all? Oh, okay. Oh, I did. Okay, look. So you can take a look at how it's graded, where y'all get that information from. So you can take a look at that, see what I'm gonna, how I'm gonna grade it, stuff like that. So this may, be um, if you are like many people, and when your professor says, I have these exercises for you all to work on your own, and you go, okay, solid, and you don't rush right off and you do those, then this is now the time to do them. All right. Like it's probably like your last chance to do them and not feel like you're going to have an absolute heart attack, not knowing what you're doing. So, review everything that's in this section, review this lecture, review this, reminders, tips and tricks, story map practice. It's got a bunch of different links and stuff like that in there. So take a look at that if you haven't already. And just let me know what's going on. Okay. Yeah, just let me know what's going on. If something, I know everybody's kind of struggling right now. So, um, Sunday it is as of right now, just so I have something to look at. Okay. Okay. Any other questions that y'all can think about right now? Okay.
So basically just follow from the top to the bottom
Mhm.
in order to fill out the rubric. Like on the story map section of Moodle, just click every link and then that'll help us fill out the rubric.
Yeah, so the rubric is just like the way that I grade it, right? And so you're just
Okay.
like, so I'm grading it based on language, I'm grading it based on map quality, I'm grading it based on, it just tells you like what I'm looking for. Like I'm looking for content, I'm looking for style, I'm looking for flow. Like it has to be good, short, not too texty, good pictures, clear images, solid, helpful, interesting maps that help tell your story, right? And so when you open up that rubric, you'll kind of check it out and it'll just be like, oh, this is this is how she's going to grade it. Um, and if you're still confused about how you actually build your story map, then you're going to want to go and do these exercises and you're going to want to review this ArcGIS story maps. And then this lab 3, was where we actually practiced using ArcGIS online. No, Taylor, I can't give a paragraph count necessarily, but remember it's a story map, so just like a long block of like paragraphs of text, it's just not a good flow. And so really what you want to think about like targeted points and subsections and headings and visual aids to support telling your story. And so visual aids can be figures, visual aids can be pictures, visual aids should be at least a few maps. So Sure, a paragraph, but like be careful with your language. Like it doesn't, if you're making, solid small points, you can do like a sidecar situation, or you could do that I can't remember what they call it, where you like swipe through things like that. Just like big blocks of paragraph, narratives are not great for this medium of communication. Okay.
So it's like a rough draft basically on story maps.
Mm-hmm.
Cool, cool.
So you just have
Like,
to press publish. You just have to make sure you publish it
okay,
or I can't see it. And so that's all. You don't have to like, it's not like I submitted it, or there's nothing you actually have to do on Moodle. You just have
okay,
to push publish, and then I'll go in and I'll check it and I'll look for them and I'll see it.
okay, cool. I was wondering what
Yeah, yeah.
what that was.
It's just an empty. Yeah, yeah. You just press publish, and then it's post, and then it's viewable. And then I can check it out, because I mean a lot of times, which is why I want it to be turned in early, because I can grade these and look at them in real time, and certainly review them in real time, like while we're sitting down next Tuesday. But if there's some sort of a simple problem like permissions or group settings or like anything weird like that, like we want to figure that out before we're sitting down. So
Okay, thank you.
sure, sure.
Around how many maps would you say? I know it's probably in
Great. At least
grading.
and that information is in this PowerPoint. Here, do we need to just look at that real quick? Oh. But I say it has to be, it's either 3 separate maps or at least 3 unique pieces of data displayed skillfully on one map. So a minimum of 3.
Oh, okay.
Yeah, yeah, and it's I'm in the
So
wrong.
if we have multiple data sets that like we're representing like all in one map,
Mhm.
but like if you make it look good and stuff, like that's
Sure.
okay.
Yeah, yeah,
Okay.
for sure. They don't, yeah. And so just skillfully displaying them, and so what I really want here is I want again, I just want y'all to go beyond like historical range and current range, right? That's so it's not unimportant. It's not an unimportant thing, but it's not where we want to go with this like conservation issue necessarily. But if y'all just review this lecture, the details of the assignment are in there. And remember, I mean, you have to go way back, but remember that like second week where we went through all of those story maps together. Um, and I showed you the one that I liked the best, and you all showed me the ones that you liked the best, and it probably was less relevant then than it would be now, but you can go through and look at some of the past story maps and see how everybody does. And generally speaking, folks in this class do well. That story map grade is gonna, I mean, y'all are doing great. I don't mind teaching GIS on an online format. I didn't think I was gonna like it, but it's pretty good. I think it would be different if I had never met a single one of you. I would like it less, but it's been pretty good, and I think y'all are doing well. But regardless of that, story maps are usually a place where people, if they try, they bring their grades up. So I think it'll be good. I hope it'll be good. Okay. What else? Any other questions? Okay. All right. Get ready for a little scrambling. So today for our final lab, we are going to work on something called interpolation. And we are also going to learn how to run some pretty interesting spatial statistics using raster data. So when we did lab 6, we created raster data sets when we were creating those um, kernel density estimates. The resulting data set was a raster data set. And so it provided some estimate of like density of mad rabbit attacks per month, right? And so just basically visualizing how something spreads, right? So that was the first time that we were working with raster data. Today, we're going to work a lot more specifically with raster data. So the first thing that we'll do is we're going to go ahead and we're going to just start from here, come here, you little stinker. We're going to make our folder. Anybody? This is us. This is us. So let's make our folder for today. Okay. And then let's go into Moodle. And in Lab 7, let's just download this folder. You've got 3 pieces of data. You've got something called 2020 hydro zipped up. You've got basins coastal zipped up, and you've got levees zipped up. We're just going to download that folder. Make sure you put it where you want it. I always just make the names shorter whenever possible. So now when we expand this Lab 7 folder, we have zipped up folder. So let's go ahead and do and extract everybody out, just so it's done. Okay. And what I'm actually going to do right here, so this is my zipped folder. This is my unzipped folder. I'm just making my names as short as possible. I'm just going to name this 7_data. And then opening this up, you'll have 3 more folders that are all zipped up. So let's just unzip those. I did that already. Okay. And so just giving all of these a chance to catch up. Okay. And then if you haven't already, download and open the lecture for today. And then actually what I think I'm going to have y'all do is jump to slide 23. And we are going to start the download for coastwide vegetation raster data. So we have worked, the data set that we're going to be working with today are the 3 data sets that we downloaded already that I put in Moodle, and this data set. So this is the same coastwide vegetation data set that you've already worked with, but you all worked with this in polygon form. So it was a shapefile, it was a polygon. It's the thing that you used for map assignment 2 that gave us fresh, intermediate, brackish, saline, swamp water, right? This time, this is the exact same, it's the exact same data set, but instead of being as a shapefile, it is a raster. And so raster files behave and look different than shape files. Remember, rasters are contiguous, continuous data sets. And so basically you have an area, and over that entire area, there's only kind of one thing that it's asking you, but it's, over this entire data set, right? And so this whole, over the whole coast of Louisiana, put your finger anywhere, and is that fresh, intermediate, brackish, saline, right? And so this data set is a continuous data set that describes marsh type, right? It's big. So we're going to go ahead and download it now because it might take a minute or 2. So in here, this is 2023 master plan. We don't want this one. We want the thing that it's going to link you up to immediately is going to look like this. We'll go to the 2017 master plan. And we're going to cruise down to coastal vegetation. And so coastal vegetation, notice that this is a raster data set. All right. When hopefully y'all have played with this or at least looked at these. I know you've looked at this website a little bit because I've made you for the last map assignment, but look at all the different things. Look at all the cool things that we have. We have restoration projects, the location of those. We have structural projects. Those are things like levees, rock walls, erosion protection. We have land change. So remember how for map assignment I asked y'all to compare those two time periods. This is a data set that does that in a slightly different way, right? So you all had to kind of do the background work on your own. We've got flood depths, we've got economic damage, we've got good old coastal vegetation, and we've got this cool data set that looks at social vulnerability to flooding. This is really the only data set of its kind in the country. It is a really cool, novel technique trying to, and this is really interesting here is the social vulnerability index, right? Because what we're trying to do is we're trying to understand how ecosystem resilience translates into community resilience and economic resilience, right? And this is just kind of a first, we're not great at this yet, but this is our first step, sort of trying to figure it out. And this is something that other states in the United States are trying to do, right? And so they're looking to Louisiana for lessons about this. And this is, so getting to know this data set will be really helpful for you, even if you don't stay in Louisiana. So anyway, let's download it. It's a big old data set. It might take a minute. Raster data sets are big. This is one of the reasons why if you choose to use a raster data set for your story map, you have to convert that raster data set into something called a tile package. This is something that I would prefer that you figure out before the end of classes, so that if you are a person that's working with a raster data set, I can give you guidance on that before you have an absolute panic attack because you can't get your raster data set into your content on ArcGIS Online. Okay, so 2017 master plan vegetation as a zipped folder, save her. Again, depending on where you are, depending on the internet, it might take a minute. So we're just gonna let that, oh look, no, didn't take any time at all. Goodness. Maybe it takes a minute to extract. Maybe that's what I was thinking. I'm gonna unzip it. So mine is up here, unzipping. That might take a minute, but then at least we have everything. Okay. Yeah, I think this might be what takes a minute. Okay. Yeah, it's the extraction that'll take a couple minutes. So while your master plan vegetation data set is extracting, we will go ahead and do the lecture part of lab. Okay. So we are going to be doing something today called interpolation. Interpolation is often used interchangeably with the term extrapolation. Inter is inside or between, right? And so really it's like between points is what that kind of means. Extra is outside of points. So think like extraterrestrial versus something else. So what we're doing here, let me actually make this look better. So what we're doing when we run these more complex spatial statistics is we're doing something called geostatistics, right? And so what this does is this uses statistics to essentially assign values to spatial, geographic areas where we don't have any data or we don't have a lot of data. And so thinking about something pretty straightforward like this is a map of a flying fox. Here we have point data, right? These are observations, occurrence records of where flying fox occurred on the east coast of Australia. And so this could be analogous to many of you used GBIF data, right, for that one homework, and this was occurrence data. So when we get occurrence data, we, go out, and something like that could be a point count survey. You could look for scat, you could set up game cameras or something like that. It's just a point in time, and you know that an organism occurred there at that point in time. You don't know how many they are, you don't know any of that stuff, but you know it was there, right? And so what we have to do is, in order to create things like range maps and create things that are actually, useful for managers and useful for ecosystem enhancement and restoration and protection and, protecting habitats, all that good stuff, is you have to figure out, what does this coincide with? Right. So I really like this kind of flow chart, right? And so what we have here is we have an occurrence of a species of interest, and then we need to know something about that habitat. So this is the 4900th time where I'm talking about habitat in this course, but really it all starts with habitat. And you figure out, are there some patterns that you can glean from these occurrence data based on environmental conditions? So what we do is what typically happens when we're trying to characterize the quality of a site for a particular species, and to determine if there's anything else that's needed to improve the quality of that site, or to make a migration corridor, or really to do anything for your organism, is we start by collecting abiotic and or environmental data. And so you go into an area and you're like, start with your heavy hitters, start with precipitation and start with temperature. Then you could talk about soil type. Then you could talk about vegetation density, vegetation height, vegetation type, lots and lots of different things. You have to know a little bit something about your animal, right? And then you can go out and you go and identify the animal, you map the known occurrence of species, and then you overlay that known occurrence of your species with your observation, with your environmental layers. And then this is where you start to create a model, right? You use algorithms, you use math, you use GLMs, which are generalized linear models, which are the models that we use to determine presence or absence. You might use a regression model if you were trying to predict abundance. That's much more difficult with species. This is where you start to say, okay, based on these environmental conditions and the number of occurrences that I have, I can say that the probability of finding an organism in similar environmental conditions is some number, right? So we're characterizing a site, and then we're extrapolating that the likelihood that your species will be somewhere else based on its similarness or differentness from where we found it before. It's kind of like applied common sense, right? Like I always find buttonbush in wet environments. So I'm going to associate buttonbush with wet environments, and therefore, if I'm in a wet environment that has similar temperature, I would assume there to be buttonbush as well. And if there's not, there's something else going on that's preventing it, as an example. What this does is this takes something like this, where we have observational point data, and it transforms it to something that we're more familiar with, particularly as we're looking for species ranges. It converts it to a continuous surface that gives us likelihood of occurrence, right? And so we get something smooth. So we have, a high likelihood of occurrence along the coast, reduced likelihood of occurrence as you go inland, right? And for many types of data, it's better to have this continuous surface data set than it is to have discrete points. Because let's say that somebody bequeaths a huge swath of property to a conservation organization, and this conservation organization has these missions, and the mission is to restore populations of gray-headed flying fox. And so you're like, you get this job, and somebody's like, hey, Carl, my guy, I need you to give me an estimate of how long it will take you to improve gray-headed flying fox populations in this area. And so in order to do that, you should be like, well, how suitable is this habitat for this organism? You know, what have other people done? What did they find? Why is it less suitable than some other areas? You know, go in and do your little ecological detective work and figure out what's going on. And so really this is a fuzzy image, but you get the idea. Building a model, right? We get these species occurrence points. We figure out some suite of environmental predictors. There can also be important biological predictors as well, but those are much more difficult data sets to come by. And then with these things combined, then you get this predicted suitability, the habitat. And so the other word for this is a habitat suitability model. So habitat suitability models, hang on a second, I just want to go, because this is something that they're going to ask you about doop in capstone for sure. And so look, I'm just Googling habitat suitability modeling in R, habitat suitability modeling ArcGIS, habitat suitability modeling in QGIS, habitat suitability mapping. A habitat suitability model is a statistical tool. And so a statistical tool is a fancy word for an equation, right? You have an equation, you have variables that you will need to solve for a lot of time. And so let's look at a habitat suitability model. I wonder if they have, wait, elm, what's your species like? Elm ash borer? Let's see if they have one for it. Oh, they do. So they have so habitat suitability models, and this, so I'm just, using Lawrence as an example. A habitat suitability model for emerald ash borer just primarily identifies areas of high abundance of ash trees. That's like literally all you need to know. Like if there are ash trees, then the likelihood that it's suitable, that the habitat is suitable for your ash borer is high. If you have a high abundance of ash trees and it's subjected to pollution, so that's another habitat layer that you could add. If you have a high abundance of ash trees and you have warm summers and cool winters, right? So you're losing those extreme minimum temperatures or something like that. And so this is, but these are, this is a specific equation that has variables that you solve for by collecting data, and some of these data need to be field collected. Some of these data you can collect remotely via GIS. Some of them you can do literature reviews, all kinds of good things like this. But it is an equation that is specific to your organism of interest that tells us how suitable a particular habitat is for your species. And what interpolation does is it basically takes that equation, that habitat suitability equation, or any equation. This can be like erosion potential. This is the same thing that's used with watershed modeling. This is the same thing that's used with, estimating temperature change or hill shade and slopes and all kinds of things like that. But basically you take your equation that is specifically tailored to your situation and you evaluate that equation across every pixel on your map. And so a pixel is your grid, right? We have our lines of longitude, we have our lines of latitude, and then we have some resolution, right? Is your pixel resolution 100 meters by 100 meters? Is it 500 by 500? Is it a kilometer? What is it? Just, whatever that is, is relevant. That's a decision that you have to make as a mapper and a ecologist. And then it just applies that equation and that algorithm over some space. And then you come up with this beautiful and sometimes helpful model that predicts the likelihood that you will find a flying fox here, even though nobody's ever seen one or documented seeing one right there, right? Interpolation and habitat suitability models and lots of different types of models allow us to predict the likelihood of some outcome given limited data, right? It's impossible to sample everywhere in the world, right? So this is how we kind of negotiate around that. Um, habitat suitability models for all of your species of interest, they exist in some form or another, unless it's a very uncommon species, something that you don't know about, right? Or it's like a new one or something. There's lots of habitat suitability models for invasive species, because as you can imagine, that's really important to understand. There's habitat suitability models for game species, recreationally important species, commercial species. USGS, so this is the agency, the federal agencies that primarily is tasked with creating these habitat suitability models. They're moving towards something called species distribution models, which are almost the same thing. They're a slightly nuanced differences, but so you could look for habitat suitability or you could look for species distribution, because think about this from an ecological perspective. The distribution and abundance of individuals of species is a direct reflection of the suitability of that habitat to support those species. So they're very similar. There is a difference between interpolation and extrapolation. Interpolation is where you estimate the likelihood of some event happening, or you estimate the number, right? And so like the number of something, would be like if you're estimating density or weight or length or whatever for your organism. Interpolation happens between two observed points, right? So let's say that we were trying to estimate leg length in green anoles based on size of species, dominant plant species. This is the thing that they've looked at, where like if it's if your plants are dominant, if they're like little weak, crappy little limbs, then your leg length for anoles is going to be a lot longer. And if they're sturdy limbs, they're going to be sturdy plant limbs. They're the anoles legs are going to be shorter and stronger, so they can jump. But so let's say that it's something like leg length, right? You have a length of a knoll or size, a weight of a knoll or something like that, right? So let's say that this is weight, let's say that this is leg length. Interpolation would occur if you have between your minimum and your maximum values. And so, for example, consider climate data is probably a more reasonable example of this. Historically, in the month of November, the minimum temperature in Louisiana has gotten this low, and the maximum temperature has gotten this high, right? If we were trying to predict the temperature in, next year, and we were interpolating, it would be somewhere between these known values. But those of you that know a little bit about climate change know that that's not exactly what happens when you're trying to predict how climate change happens, because what we're doing is this is why when climatologists say like, we are experiencing warming that is beyond the bounds of any normal or natural variability that we've seen. This is what they're talking about, is they're saying, historically, we never had an upper range that was here. And every year it creeps up this line. So what we need to be doing is we need to be extrapolating our future climate models so that they can actually predict values outside of the range of sample data. Because every year it gets a little bit hotter, the mean minimum temperature gets a little bit higher, winters are getting warmer. That's a really, really big deal. And so if we restrict ourselves to just between the range of sample data in history, then we're not going to be able to accurately predict how climate is going to change and how climate is going to impact species and habitats. So often these terms are used interchangeably, but they're not interchangeable. They're different. And a lot of times if you were looking at these data, they'd say, oh, well, you never should extrapolate your data. Don't ever extrapolate your data. But it really depends on your data set and what you're kind of trying to explain, right? Sometimes extrapolation has to happen. But remember, extrapolation just occurs outside the known range. So it can be under the minimum, over the maximum, something like that. Interpolation stays within the known range based on the data that you have. A couple different interpolation methods that we're going to have. We're going to play around with something called inverse distance weighting. And so inverse distance weighting, basically, you have your pixels And you have your individual, like your grid across your map surface. And it says, okay, like based on and I have a bunch of different observational points, like let's say it's, density of species or presence of species. In this case today, what we're going to be working with is salinity. So let's say that this is the center of your pixel for your map, and you have this salinity value that is 0.03, and this salinity value that is 0.04, this salinity value that is 0.03, and then you have all these out here that are 0.25, they're higher or something like that. What IDW does is it weights those observations that are closest to the center of the cell higher. So it basically just think about like the way that you're graded in, not the way that you're graded in this class because we just do points, but like for ecology or something like that. For ecology, I give y'all a couple of homeworks, and those homeworks are worth like 10% of your grade total. But your first test is worth like 12.5% of your grade total. So your first test is weighted higher, right? So it just means like in this case, these are weighted higher. They have more value. This is an interpolation method. It doesn't do great in areas that have a lot of change, particularly a lot of rapid change. So like highly dynamic environments in terms of salinity, highly dynamic environments in terms of elevation or temperature or, soil moisture potential or percolation rate or anything like that. In areas that have a lot of rapid change, this method doesn't work very well because basically it's treating these values that are further away from the center of your pixel as less important. When they really may be equally as important or to the ones that are in the center. Natural neighbor is an interpolation method that's kind of similar to inverse distant weighting. Natural neighbor and natural neighborhood statistics are like a family of statistics that we'll use a lot, where basically, we have some observational values. We have some polygons. In this example, it's using polygons. We have some points, in the center of those polygons, and what natural neighbor does is it basically it creates these new polygons, and it steals the value of the neighboring polygons, and it's going to weight whichever polygon section is like bigger in this new polygon that they have formed more heavily. So it's the exact same equation as inverse distance weighting, but it creates these kind of funky polygons across your map instead of creating just like a straight up raster. And it allows extrapolation as well as interpolation. I don't use natural neighbor a ton. Some people do. Again, it just the right tool for you for interpolating. It depends. It depends on what you're trying to do. So it's kind of up to you. Spline is one that we're going to practice today. So spline, think about the spline and Krieging are the two that we're going to talk about the most. Think about spline and Krieging as if you were trying to stretch a flexible rubber sheet across an uneven surface and like stick it in and make it as smooth as possible. So if you're trying to make like a face mask or a face mold or something, right? You take this, sort of flexible surface and you push it into your nooks and crannies, and then you pull it off. And that is a model of your face. That is an abstraction of reality. It's not your face. It's not totally right, but it's as close as it can possibly get. What spline does is spline requires that any observational point across your spatial surface is kept as true. So the sheet must pass exactly through these observational points, right? It must contain, so here we're looking at an interpolation. We're looking at a model, right? So if you've got some value here, if your distance was here, maybe you didn't sample data right there, but you're going to come up with the value, right? Based on this model that has been made, right? These are your observational points. These are your assigned values in red. This is your model, is the black line, right? Same thing projected over three-dimensional space, right? We have the observational points and we have the model across it. Spline must keep the assigned values. Kriging doesn't have to. Kriging basically, what it'll do this thing where it will sacrifice passing through the exact assigned values if that means creating a smoother surface. And so one of my GIS wizard friends, he sometimes has problems with spline, especially when it's looking at really detailed monitoring data where, or I'm sorry, Kriging, where Kriging tries to be so smart that it actually ends up being very silly, which is like, welcome to academia, y'all. Like, that's what it's like. Everybody trying to be so smart that they end up being silly. But so compare the way that this model looks when you're looking at it just, you know, as a line, right? We have our observed points. Kriging makes that line as smooth as possible. And it only passes through one of two, three, one, two maybe of these points, right? It is not honoring those assigned values versus spline, where spline has to pass through those assigned values. And so for you, right, like the difference between using spline and using Kriging to interpolate values across the surface really depends on your confidence in your input data. So like if you're assigned values, you're like, these are so, I know that this is right. I measured the diameter at breast height of this tree myself. I know that's how big it is. So I know I have a couple of people that are doing forestry stuff. And if you haven't done it yet, you will. But let's say that you go out with a team and you separate. And you know that one person on your team is really good at estimating tree height. But one person on your team is not good at estimating tree height, right? Like you're like, oh, buddy, you know, we've got Demarco and her depth perception sucks. She can't estimate how tall anything is. This is a real thing that happens with foresters. Some people are great at estimating the actual height of a tree. Most people are not good. It's hard to do. It's really hard to do. But you have to make that decision. You're like, okay, if I'm trying to make a model of canopy height given environmental conditions, you know, do I make it spline, right? Do I make it pass through those assigned values because I have faith in my team? Or do you say, no, we can go ahead and use Kriging because, you know, the variability is so high and uncertainty is really high or something like that. So the method that's best depends on you and your data set, depends on your best professional estimates, and they all have different benefits depending on what you're doing, right? So IDW sometimes is great. Kriging is sometimes exactly what you need. Spline is sometimes the best choice, right? It's the best tool to complete your task. It's variable. Spatial statistics. So we're, you know, so thinking about interpolation is making a model, right? And the example that I've used for this model is a habitat suitability model or a spatial distribution model. Can be any type of a model. And so it's definitely working in statistics, but these are more straightforward kind of operations in spatial statistics. And so you can have some spatial statistics that operate just on a single cell. It's a per cell function. You have one raster data set, we have another raster data set, and you do something with, you overlay those raster data sets and you multiply them together or resubtract them. And so a lot of times I'll have to do this, like if I'm trying to come up with a difference map. So like if we have probability of occurrence for one time period and probability of occurrence for another time period, I might want to know what that change in probability of occurrence is. Like, did it go down? Did it go up? Did it stay the same? Was it within some range? And so that's something that I could do on a per cell function. I would just subtract one absolute value of one layer, one raster data set from another. Maybe I have a data set that I need to make a little bit more general, right? So that I need to, you know, create a raster data set where the output value is not just a function of the pixel directly on top of it, but it's a function of its neighbors. And so something like this, these are known as neighborhood operations or neighborhood statistics, where basically you're like, okay, I have a coastal data set and I have my resolution is 500 kilometers by 500 kilometers. But I want there to be, because this is a continuous data set, I want it to be like a more, smoother than that. And so the example that we're going to use today for these focal operations is we are going to take land and water. We're going to have a data set that's land. We're going to create a water data set. We're going to create a land data set. And then we're going to try to calculate edge. And so you're not just doing land over water. You're calculating, this is a land pixel, but it's surrounded by water. And so basically estimating like the relative like edginess or wateriness to it, right? And so this is something that we have to do a lot in ecosystem science and in landscape science, because of its importance with evaluating edge, because of its importance in understanding how neighboring habitats influence some core habitat. And so these are focal operations where you can kind of focus on different sizes based on whatever you need based on what you need. Zonal operations, you can define these big zones that you want to create raster data sets for. And you can also have global operations where you use like really, really great big global statistics, right? So this is kind of a question of scale, right? Spatial statistics, is it teeny? Is it per cell? Is it focal? Is it like an area, one cell and it's one pixel and its neighbors? Is it bigger? Things like that. Okay, so what we're going to do today is, I actually don't think you guys have this or need this. Yeah, you don't have this. So you're going to ignore this slide because I don't even think y'all have it. So here we are. We are going to start here with these lab directions. So what we're going to do, I'm going to move this over. Let's go ahead and open up Arc Pro. Where is she? And hopefully everybody's coastal vegetation data set has downloaded at this point. Is anybody having trouble with that? Is everybody good? Come on, buddy. You'll be okay for a minute.
Mine just finished downloading so that I could put it into my file. So now it's just starting to extract. So I'll just try to follow along
Yeah,
mentally.
it's okay. We're not going to use it right away. We're not going to use it right there. We're going to use our basins file and our coastal vegetation file we won't use for, so y'all have some time. We're not going to start with it right in a second. So don't worry yet. Okay. All right. So we're going to open up a new project and this is going to be lab 7 project. Okay. And notice where it's putting it. We leave it there. That's not where we put our shape files, even though it tries to. I'll give it a chance to open. So what I'm gonna have y'all do for this map is I just wanted to show this to y'all. You can set, if you know that you're gonna be working in one area for the whole time, you can actually set your coordinate system for your map, right? Instead of just setting your projected coordinate system for each individual data set, which we'll do anyway, but you can actually set your coordinate system for your map. So we're going to go ahead and do that. So once your map opens, go up here to map, right click on map in your table of contents and go to properties. Once you're in properties, go to coordinate systems. And what we're going to do here is, you'll see what it's in. It's in WGS 1984. I'm going to go ahead and expand Actually, I can probably just do this. Let's just go to projected coordinate systems. We're going to cruise down to UTM. We're going to cruise down to WGS 1984. Expand Northern Hemisphere. You've been here before, right? Good old WGS 1984 UTM Zone 15 North. We do this often, right? But we have never done it before by setting the projection for our entire map. And I just want to, I want to do that because I want to kind of show y'all how it's going to change the way that your map looks on a flat surface. So I'm going to select WGS 1984 UTM Zone 15 North. And I want y'all to either watch my map or watch your map as you hit OK. It's going to flash. And then it kind of goes whoop. Yeah. So subtly different, but different, right? So this is telling us that, you know, this transformation has happened, right? We are in the right projection. The model that is estimating where these points should show up on a flat surface based on where these points are in the real world works best in this area, right? Right in the center. Okay. So now let's expand catalog, and we're going to connect to our folder. Right click, add folder connection. Navigate to where your stuff is. And then once you're connected to Lab 7, you'll have two files. Well, you might have two files in here if your master plan vegetation is finished extracting, but you're going to have this data file. This is the stuff that we downloaded from Moodle. And then you'll have 2017 master plan veg. That is what you downloaded from CPRA's website, if it's gotten there yet. But we're going to start here. So go ahead and expand your data. And we're going to pull in this coastal basins file. So open this up. Notice we've got a lot of different things. We've got a KML. We've got a shape file. We also have this layer file. Let's just grab the shape file. Basins coastal shape file. You can see by the symbol, this is going to be a polygon. Pull it in. Make sure you put it on top. Okay. So in South Louisiana, we organize our coast into these things called basins. Each of these separate polygons that you're looking at represents some suite of environmental characteristics that are unique to these regions. So this is something that's really useful to do in dynamic areas, particularly in dynamic coastal areas, even more particularly in estuaries, because we have a lot of different things going on, right? So I'm just going to turn this off. Don't worry about it too much, but we've got different types of water bodies. We've got different amounts of freshwater flow. We've got different marine influences. We've got different rates of subsidence. We've got different rates of sedimentation, right? Organic matter accretion, all these kinds of things. And what research over many, many, many years has shown is that we can create these meaningful classifications and meaningful distinctions across our coast with these coastal basins. Okay, so what we're going to do here is I want you all to look at your attribute table first, because this might be something that you would use another time, right? And so let's say that you just wanted to look at one of these basins. You were like, I want to, let's say that you really loved map assignment 2, and you were like, I want to talk about coastal change, but I really want to focus on Terrebonne Basin or the Bird's Foot Delta. And I really want to look at those marsh types there or something like that. You might want to you would just extract just that basin. But let's look at our attribute table any minute now. There she is. And so our attribute table for coastal basins is going to be pretty straightforward, right? We've got 10 separate basins described by, some dominant feature where they are. We've got size in acres, we've got size in square miles, we've got length of the shape and area of the shape, right? And we have this, so all files will have this FID thing that shows up. So what we're actually going to do first is we are, oh, I'm sorry. Let's pull in our CRIMS file too. So we're going to go ahead and do this again, just in case y'all didn't last time. It's a good memory. Remember how you get CRIMS data when they're downloaded. You get one that's just the coordinates, and then you get one that is the actual information. So what I'm going to do here is I'm going to take this CRIMS coordinates. I'm going to export that table to point feature class. You could probably go in and find this somewhere else. We do have it. I think we made it in lab 5. So you might have it in lab 5, but I'm just going to remake it because normally it doesn't take very long, but the computer is really slow right now. I just goodness. Goodness. Okay. So output feature class, remember, always check and see where this is going, because this is just trying to put this in our default folder, right? I'm going to put this in lab 7. This is going to be CRIMS XY GCS, because remember, you just give it a geographic coordinate system the first time you pull it in. X field is going to be longitude, yes. Y field is going to be latitude, yes. Coordinate system, GCS, WGS 1984. That is just a geographic coordinate system. That's exactly what I want. If yours does not say the same thing, you'll expand this and go hunt down geographic coordinate system. Just go to run. This should be review. Y'all have done this before. So now we have CRIMS. If you open up your attribute table for your CRIMS stations, remember, very simple. It's just your station ID, your longitude, and your latitude. Okay. So what we're going to do now is we're going to join it. Remember how to do that? You're going to hop up here to your table of contents. You're going to go join and relates. Add join. You were going to add this CRIMS XY based on the station ID to this other CSV file that comes packaged. Remember, this guy's just your XY. That's the stuff with your goodies in it. We could go in and look at it as a CSV, but we're just rocking and rolling. Join table field. You could validate your join. You can always scroll over this little exclamation point. It increasingly likes to give this warning where it's like, we have to index your stuff. It's fine. You could do it or not, but we're fine. We're just gonna go forward. Let's go to OK. Remember, joins are not permanent. All you have to do if you screw up and you do the wrong thing is remove your join. But if you're not organized in your brain, it might freak you out with what you're doing. So here we've got everything joined. We've got all kinds of good information.
I have a question. Whenever you're using, so I know like right now we're kind of using habitats, which is kind of hard because you would need latitude and longitude. But if you're doing building data, would it be fine if you just had an address and it like would still show up on the map, or do you still need that latitude and longitude?
You're going to, if you can get it to show up on your map first, great, go with that. But you always want to go ahead and add that geometry. You would just like add
Okay.
and then add that, because Just think when you're moving data sets around, it's just so much easier to display your data if you just have a lat long, especially if it's like a point.
Yeah. Is there, okay, can you do a quick like a redo of how to do lat and long? I know how to do the hectares and acres, but I'm not, is that the same thing? Do you do the same?
No, you have to add a field. And so you go add, and that's in, I can't remember what lab, I think that's lab 3 as well, but you go to add, and then you would just name it and tell it how many.
Is that when you do the pull down of like double and single?
And so you have to
And
add.
which one did you pick?
Huh? Float.
Which one? I'm sorry, I cut you off. Oh, float. Okay.
Mhm. And yeah, and that's in a lab somewhere. I think it's lab 3. So I think it's written down in lab 3. But if you need help with that, we can look at that like next week when we go over your math assignment. What's up, Stephen? Stephen, you got your hand up. What's up?
Oh, I'm sorry. I think one of the longitudes is messed up on the Crim station. Or yeah. One of them's it's backwards, the longitude and latitude.
Not on my data set. Did you are you using the same data set that I am?
I think so.
What? No, you're right. Okay. So remember how this happened the first time? Ah, shit. Okay, so let's delete this. Ugh, so we got to open up the CSV. Actually, so let's see. Let's see if instead of this, if
It's just
we're
one
all
of
good,
them.
if we're all good. No, it's this happened like a million years ago. If we're all good, we remember where this Crim's XY data set is that we originally made. Do you think we all remember? Do we all know where that is? Should we try it? Right click and go to add folder connection. Let's go to lab 5. And if you expand, and if we expand lab 5, everybody should have this Crim's XY GCS. Does anybody not have that? If you don't, we'll all go through and we'll open up the CSV and we'll switch the latitude and longitude and we'll remake it. But if we can all find this.
It's in the data sets for lab 5.
No, I mean, that's not where mine are. But it should have a name, something like this, right? Crim's XY GCS. That's the one that we corrected in lab 5, I think.
It's also the one we use in map 2, right? The map assignment 2.
But it should just be, so basically what you should have is you have this one Crim's data set that just it has that same data. It just has latitude and longitude. All correct, right? Yeah. Can everybody find this? Everybody have this? So yes, everybody's got it. They may not have it.
I can't find mine.
Okay. All right, we're gonna go ahead and remake it. Okay.
It's under
Okay.
11 or yeah, 11,028 Mars class under data set. That's where it is for me at least.
Everybody's gonna have their data in a different
All
spot.
right, never mind.
Which is what it certainly sounds like. Okay, so we'll just fix it, and this will be a good review. So we're gonna open up lab 7 in File Explorer. We're gonna open up the data. Oh, you know what? I'm gonna go. No, thank you. We're gonna open up this coordinates file. And you know what I'm actually gonna do is I'm gonna go ahead. Where did I? Oh, did I delete it already? I deleted it already. Huh. So when you open up your hi, honey, sweetie. When you open up your CSV, for your 11133 coordinates, right? You are just going to select everything, go up here to sort and filter, go to custom sort, and you're going to sort by, it doesn't matter, you can sort by longitude or sort by latitude. I'm just doing longitude because it was on the top. And then scroll all the way to the bottom, and that's going to be where our error is, right? So we've got this 29.896. Just going to copy that, drag that 92 over. Great. You're just switching them, but it always takes longer than seems reasonable. So we're just switching. If they just got the latitude and longitude switched at some point when somebody was doing data entry, it got switched. And it hasn't been corrected. So now that we have that latitude and longitude corrected, everything should be all good. Just go up to the save it, close it. It's going to fuss at you. It's going to say, are you sure you want to save? Oh no, it doesn't fuss at me anymore about saving it as a CSV. Okay. Now, just to be careful, I'm going to refresh my Lab 7 folder, and I'm going to delete this CRIMS XY GCS that I made a minute ago because it had the point in the wrong place. So, okay. All right. My computer's struggling a little bit. This is a fun thing. Okay. So everybody got their lat long switched. Now we'll do that step again. Under your coordinates CSV, right click, export, table to points. Since we deleted the thing that we had already named CRIMS XY, we can name it the same thing, CRIMS XY GCS. X is longitude, Y is latitude, coordinate system GCS WGS 1984, ready to go, and just hit run. Wonderful. Go ahead and right click and go zoom to layer. And now everything is right where it should be. Open up your attribute table for this, just to check and make sure. Yeah, looks good to me. You know what, y'all? Let's do something else. Since we're here, go ahead and open up that 2020 hydro folder. And I want y'all to open up the one that just says 11133. Because I suspect this is like a buggy thing with ArcGIS. And where if we open up the CSV and we sort it based on something, then it imports better into GIS. Then we don't get that like weird thing where it's not reading our columns as text instead of reading them at numbers. Remember how some of y'all had that problem? It was really frustrating. This can help. So let's go, we're just going to select everything. I want you to go up here to sort and filter, and let's sort and filter custom sort. Doesn't really matter what we do it by. Let's sort it by max salinity. Great. Then that's all we have to do. fine, no problem, go to save, close it. I'm just interested. I don't, hopefully we don't have to do that with every field because we're certainly not going to do it, but that should like trigger everything to work properly. Okay. So now we're back on Arc Pro. Right click on your CRIMS GCS XY. And you are going to scroll down to, what are we doing? To joins and relates. We're going to go to add join. Joining it based on station ID. Joining it to its 11133 friend. And I have a thing that happens with students where, especially if you guys panic, a lot of times you end up trying to join itself to itself. You forget like which is which, or you try to create a shapefile out of the wrong CSV and things like that. So just try to like take it down and stay organized and open up your CSVs and then close them, right? They must be closed to do these operations in GIS. Then you have to close them, and then that usually makes more sense. Go to OK and watch your attribute table change. Wonderful. Okay.
Professor DeMarco,
Mhm.
for the input field, I don't have station ID on.
Do you have something like site ID?
Is that the same thing, site ID?
Mhm. But I'm
Okay.
wondering if you're in the wrong order.
Should I share my screen?
Yeah, hang on.
Okay. Sharing.
Okay, now you're ready.
Wait.
Can I see your attribute table for your CRIMS? Yeah, it looks fine. I don't know why yours says site ID and mine says station. That's odd because it's the same data set that we all downloaded from Moodle, but
Okay.
it should be, it's the same thing. That's very weird.
I don't have 1113.
Yeah, you're not gonna have it there because it's not in your, it's not on your
Oh,
table.
so I have to click
Yeah,
it.
you have to
I
find it.
got
Yeah.
you.
Yeah, it's right there. It's right
Oh
there.
wait.
Okay.
Yeah, forget that.
Okay, you should be all set. Yeah. So Catalina, this answers your question too. See how he's doing input field is site ID. My input field was station ID. It should be either site ID or station ID. I don't know why they're saying
And then right here, join field.
nothing. Or station
Okay.
ID. That's it. Yep.
All right. Just press OK.
Mhm.
All right.
And then, yep. Oh no, you don't have any data.
Yeah.
Mine's doing the same thing too.
Okay, get out of your join. And this is the file that everything was downloaded from Moodle.
Yes, ma'am.
Okay. So go to your table of contents. Your table, that's your catalog.
Oh wait, yeah.
Yeah, right click. Go to remove join.
Uh,
Yeah.
just press OK.
Yeah. Okay. Open up your CSV for 11133.
Okay.
So like in File Explorer, yeah.
Yeah.
Everybody else do this too, okay?
Oh. You see it?
Uh-huh. Okay. All right, close it. Go to your CRIMS CSV, just the coordinates one, and open that one up again, please.
You see it?
Mm-hmm. Mm-hmm. Okay. All right, close it. All right, go let's remove this from your, so everybody do the same thing. Remove that CRIMS XY PCS from your table of contents. Why, oh, GC, yeah, remove that. Okay, and then I also want you to delete it from art catalog. Everybody do the same thing. Delana, whoever else was having, yeah, delete it. Okay. Open up labs, expand lab 7 right there, or lab 7 data, yeah, wherever your CRIMS stuff is. Okay, expand hydro. Okay, right click on coordinates. Export. Table to point.
You want me to
Yep.
do everything?
Yep, you're doing
Okay.
great. Yeah, do that. I just want to see.
Uh, uh,
Yeah, you should be able to name it the same thing. Okay. Save. Okay.
Everything looks good.
Yeah. Okay. Open up your attribute table. Okay. Try the join again. So now we have, yeah.
Now it's a station ID.
Uh-huh.
And
Yeah,
then
and just go to run.
do I have to validate it
No,
first
just go to
or?
run.
Okay. Oh, everything looks good for me, at least. It's so weird.
Did you do that exact same thing moments ago?
Yeah, I did.
Yeah,
I mean,
that's crazy. Like,
I had longitude and latitude sorted in ascending order or something like that, but
no, sometimes it makes no sense.
I got you.
Sometimes
All
it's like
right.
taking your car to the mechanic. You're like, something's wrong, and then the mechanic's like, this is fine. Like, no, it's just, like, I'm sorry that happened, but it looks right now. Delana, how does yours look?
Mine's good now too.
Okay, you can go ahead and stop sharing, Stephen. We're
All
all
right.
right. I'm sorry, y'all. It's bizarre, but sometimes it's just like stuff hasn't closed. It's it's like especially if you're working on campus or you're uploading anything to OneDrive, like the upload hasn't finished or like it just is so bizarre sometimes why this happens. But sometimes you really just have to like be patient with it, close it all down and start it back over. Okay, so now what we all should have is we have this joined CRIMS XY GCS file. Remember that joins are not permanent, right? Not permanent. So what we're going to do is we're going to wrap this up and create a nice new shape file for this CRIMS hydro data set that has a projected coordinate system. So this is something we've also done a million times, but you can never get too much experience. So go up to your CRIMS XY GCS, the one that's joined in your table of contents, right click on her, cruise down to data, go to export features, output feature class, expand this folder. Every time expand this folder. This is going to be CRIMS Hydro 2020 PCS. Kind of a long name, don't love it, no way around it. Go to save. We are going to give this shapefile a projected coordinate system. So hop over to environments. Under output coordinate system, remember when we set the coordinate system for our map at the beginning, grab your map. It's going to populate with the correct coordinate system. Go to OK. So now we have two data sets that are essentially identical, but one is a shapefile that actually includes all this information. This one is just a join, right? So let's remove this CRIMS XY GCS. Let's open up the attribute table for CRIMS Hydro 2020. And just to show you, just to kind of prove my point that joins do not persist, really not safe. Hang on a second. computer's just like having an absolute fit. Yeah. I thought so. It's in lab, yeah, so like lab 7. Oh. Don't just, I'm just sorting something out right now. Don't follow me. Yeah, it's in here. It's just taking, oh, okay, that's what's going on. So this is super boring, but you see how status over here, everything is like thinking, right? I've got pieces that are still thinking. I'm assuming that's why it's still like updating to OneDrive or something dumb. So I'm assuming that's why it's not showing up in art catalog, but it's odd this is happening. It's really taking forever. There it is. God. Okay, so to prove my point, right, this CRIMS XY GCS, that join does not persist, right? So if you're trying to submit something that has all of this join data in it, you have to actually join it and then export it as a shapefile. Okay. But all we should have on our table of contents right now are CRIMS Hydro 2020 with a projected coordinate system. We have this basins coastal, which we're going to need to give a projected coordinate system as well. We'll do that in a second. What I'm actually going to do is maybe nothing. I could go over here and clean up this attribute table a little bit, just deleting this field, right? Because we've got 2 fields that both say the same thing. One says station ID, one says station 1. You can delete it if you want. I'm just going to go ahead and delete it. It doesn't really matter. Then it's just less cluttery. Nicholas, do you want to share your screen?
Yeah, so I did the export as a shapefile with this, and
Turn on your CRIMS XY GCS. Okay, right click on it. Go to properties, source. Okay, that looks fine. Close that out.
does that need the projected?
Remove the CRIMS Hydro 2020 PCS. Or wait, actually go to properties, go to properties. No, I want to go up. I want to see where it is and what it's named. Go to the top. No, I'm sorry, in source. Right there under shapefile. Crew, yeah, I want to see that whole thing. Okay. Okay, close this. Delete the remove CRIMS 2020. Okay. Catalog. Pull that CRIMS 2020 over again.
Oh, there we go. All right.
Well, is that it?
Yep, it's there.
Turn the other, okay.
This thing is, hate this thing.
It's just, I mean, especially if you have like, I don't know. Maybe it's like hackers. Oh gosh, it's so weird, but I'm glad it's there. How's everybody else doing? Are we all here together? Okay. Okay. So now let's take this basins coastal file and we're going to export this. So just right click on that, data, export features. I'm just going to call this basins. And the only reason I'm doing this is because I want to make sure that it's in the right coordinate system. Okay. And so that I don't corrupt my original file because we're going to do some editing on this basins file. So we just go, okay. And so we have 2 identical files. One of them has a projected coordinate system. One of them does not. Right click on basins coastal. That's the original one. Remove it. So we have done a lot of data hygiene, a lot of housekeeping. We've barely, feels like we've barely gotten anywhere, but really, really, really starting and setting yourself up well from the beginning makes it so that you don't absolutely melt in a few minutes. Okay. So now we have a basins file. What we're going to do here, I want you to just select this basin file in your table of contents. And then you are going to hop up to edit on your ribbon. And then under edit, in here in these like commonly used tools, you're going to have an option to merge, right? You also have this option to split, right? So we could split these and that would give us a shapefile that had each of these separate basins as separate shapefiles, which is something that can be very handy. We're actually going to merge this file because what I want is I want a boundary. We're going to create a raster file across the coastal zone, and I want it to just be this whole boundary, right? I couldn't find this data set just standing alone by itself, so I'm making it, right? I'm making a background data set. So I'm just going to hop up here to merge. And then what I'm actually gonna, oh no. So what I'm actually gonna do is, it's asking me what I want to merge. I'm gonna open up the attribute table for basins. And I'm gonna select all the attributes in this attribute table, just because it's easier than me manually selecting them on the map, but totally would work. You can, because remember when you select something in the attribute table, it selects it on the map. And when you select something on the map, it selects it in the attribute table. So selected, it's selected across the board. So I selected all of my basins. Now it's populating this modify features, this merge tool in here. It is going to name this Atchafalaya, right? And so it says features to merge. It's just preserving. It's basically going to lump all of these together. My shape area will change. All of these identifying units will change, all of that stuff. But because we just need the actual base file, like the background, the name actually doesn't matter, right? They're all getting merged. This is ready to go. Just go to merge. And now we have this basins file that we created. So remember, our original file was out there sitting here doing just fine, right? So if we panicked, if we freaked out, if something got weird, it's still sitting there. It's just fine, okay? This new file that we've created, we could undo it. We don't even have to save these edits, right? You can discard if something went weird. You could delete it, whatever you wanted. We're going to go up to save all edits. And then we're going to go up to map and we are going to clear selection. If you have anything selected and you don't mean to, you're going to have this like cascade of problems when you try to do data analysis. So we're going to clear selection. So now we have one big coastal zone, right? Actually, that would have been a better name for this file. I should have named this file coastal zone, probably. Oh,
Can
I
you
think
go back to what you just did? I'm sorry. I got kind of lost there.
so you go to edit. You're going to
Merge.
you're going to open up, yeah, you're going to go to merge. You're going to select all of your basins in your attribute table. Actually, you know what? I'll just remake these and rename them. And then you go to edit, and we're going to merge. I'm not going to remake it because I have to change that name somewhere else. And then you just go to merge.
Okay.
Thank
you.
You're welcome.
I thought it was gonna create a whole new shapefile. That's why I was like,
No, so because you're editing, it's working on your current shapefile.
I got you.
and that's why we go through that extra step to kind of create a new one. So hang on, I'm going to try something and we're going to see if it works. I'm going to remove basins from my table of contents. Y'all don't do this yet. I'm going to go over here to art catalog. Where is the basis file? I'm trying to change the name of this from basins to coastal zone. It's just like taking forever to update. It sucks. It's just gonna make the lab, just give me a second while my computer just loses its mind. It's just gonna make the lab a pain in the butt because I refer to this as coastal zones throughout the entirety of the lab. Ugh. Ugh. Well, I guess I am gonna redo everything. I'm just redoing everything because I'm getting frustrated. It's not showing up. It's taking me long enough. Oh, can y'all, we're all just gonna remember that in the lab where it says, oh, see, now I can't even pull anything up. Okay. Wait a second. Ugh. Oh, there it is. Okay. So this is my basins file that I made that has a projected coordinate system. I'm literally just trying to change the name. And a lot of times you can do this in Art Catalog. And if you do this in Art Catalog, yes. So think about it this way. Like if I wanted to go and change the name, so I named that Basins and I meant to name it Coastal Zone. If I wanted to go in and change the name of that in File Explorer, I would have had to do that for every single one of these file extensions. Same thing like if you were trying to delete or copy paste or something like that. But if you do these operations in Art Catalog instead, it automatically updates that in File Explorer. So what y'all are going to do, if you didn't stay with me and do it already, is you're going to go over here, you're going to find your thing that says basins, whatever the one is that you just made, the merged file in your art catalog. You're going to index finger click on it until it highlights like this, and then you're going to go in front of that .shp and you're going to change that name to coastal zone instead of basins. So this is a really helpful thing to stay organized as well. Okay, now I'm going to pull it back in. Okay, so now we have CRIMS Hydro 2020 with all of this good data. We have a coastal zone boundary, essentially, and we have everything in the same projected coordinate system. Okay, so now we can move on. So at this point, we are on this slide that says IDW. Let's take a short little break to get water and go to the bathroom or whatever we need, and we'll come back in like 2 or 3 minutes and start this interpolation. Okay, we're just going to take a quick little break because this is I warned everybody this lab is rough. So quick break. Been at it for almost 2 hours. Just very, very quick. Okay. All right. Hang on. Okay. Hopefully everybody's back. Okay, so here we are. If you're trying to follow along with lecture, we are here. We're at this IDW point. Oh, and there's something that I should mention. ArcGIS and the Esri company specifically has amazing online resources. Like in theory, you could like just buy a license to using the software and then just train yourself to do it, right? You could just go through all of these tutorials and train yourself to do it. So if you are, the online resources are really amazing. AI and Chat GPT is not good for this particular program because I have students all the time. They're like, well, I asked Chat GPT and they couldn't help me. No, it doesn't work. Not yet. They're not, they can't quite do this yet. So, but ArcPro has incredible resources online. And so if there's anything that you're curious about, or anything that you don't understand, just try to ask the interwebs as specifically as you can. But we're going to go through and we're going to do 3 different types of interpolation, right? So we're going to do inverse distance weighting first. And so to do that, we are going to go back over to ArcPro. We are going to hop up here to analysis on the ribbon, and you're going to expand tools. That's also available in the geoprocessing tab over here in your pane windows. And we're going to go over here to toolboxes. And then once you're in toolboxes, you are going to cruise down to where you do all of your raster stuff. So what we're actually doing when we do an interpolation is we create a raster from some other type of data. In this specific example, we are creating a raster, a continuous surface that is going to tell us what average salinity was in the year 2020 from point data, from vector data. And so we're coming down here to spatial analyst, expanding that. You're going to go to interpolation and look at all these nice little interpolation options we have, right? All the tools, you can do all of the tools with almost no expertise, right? Like if you have the money and you have the data, you could run these tools and you can tell people anything, right? But of course that's not the way that any of this should be done, but it's the way that stuff does it. So what we're going to do here is we're going to work on IDW first. Input point feature values, very simple over here in our table of contents to start with. We only have one option, CRIMS Hydro 2020. So what it's going to try to, what it defaults to is like the first field, right? It's defaulting to this longitude. And so an interpolation based on longitude is not helpful, right? That's not anything that we would possibly want to know. There are, however, lots and lots of other fields of data in here that give us really great information. They've got average salinity. We've got standard deviation. So it's like how much this particular site, like how much does this vary away from this mean, right? Is it really, and so this gives you an estimate of variability at your particular site. We've got max salinity, we've got minimum salinity. Remember, a lot of organisms don't operate on mean conditions. Like if the salinity gets too high for a plant to survive, it's going to die. If salinity gets too low, doesn't really happen, right? It's many organisms are operating on these thresholds, right? And so like if it's too cold, so we actually have water temperature here as well. If the minimum temperature, and so this is an example like water hyacinth. Delane, are you doing water hyacinth? You are, aren't you? Pay attention.
Yes, ma'am.
Pay attention. So because you can get CRIMS data for many years, you can look at how the mean temperature, the minimum temperature has changed over time. So you could do this interpolation on minimum temperatures. And a lot of our invasive species, our climate change invaders particularly, are very sensitive to minimum temperatures, right? And so water hyacinth, it has to be below freezing for like 10 days or something to actually kill all of the water hyacinth. And so what you would do is you would, you could figure out like, did the mean temperature go below freezing? How long did it go below freezing? How many years has it gone below freezing? And you could see, what I haven't done this analysis, but I imagine what you could predict is or what you would, based on your data and your spatial analysis, is you will find that minimum temperatures are higher. So it's not freezing, as it's not getting as cold, and it's not getting as cold as often either. So you can get all of this great data from CRIMS, not as readily available in other parts of the country or the world. So we're going to be working with average right now, right? We're going to be working with average salinity. So that's this guy up here. So we are going to ask this interpolation to be run on that field. So you're going to expand this z-value field, go to average salinity, output raster. This is making a new raster file. We're going to name this something short and sweet, like IDW1. Always add a 1 just in case something goes crazy. We are not going to accept this default output cell size. What is standard when we're looking at a coast of this size, with the resolution of data we have, is you would do something, we're going to do 500 kilometers. All right. It could be 100, it could be 200, it could be whatever. Our appropriate cell size is 500. So we're going to do 500 for output cell size. That's basically, it says, I'm making a raster for you. What resolution do you want this raster to be at? It could be anything that you want, right? And so if you're getting really, really specific into the spatial components of this, you would run some pre-processing analysis to figure out what makes the most sense and what resolution is most appropriate for your study area, right? But for this particular size of a study area, these are like some standardized values, but for this particular size, we'll use 500. Okay. So for this, we're going to leave all of this stuff alone for now. What we are going to do is we're going to hop up here to environments. If you don't fool around with environments when you're creating a raster, it's going to give you a great big huge rectangle that's going to extend from your furthest points, right? It's going to go out to, we've got CRIMS points up here, we've got CRIMS points down here. It's going to give us this big square. It's going to be fine, but it's not like the prettiest thing in the world. So what we're going to do over here in environments is we're going to select the appropriate output coordinate system. And we're also going to go here to extent. This is why we made that coastal zone file. We're saying, I want you to go all the way out to the edge of that coastal zone file. And not only that, do I want you to go all the way out to the edge of that coastal zone file, but I don't want to see anything beyond that. I want you to to give me this mask so that it looks nice, right? So we're going to go to mask, and we're also going to select coastal zone. So we've got our output coordinate system, we've got our processing extent, and we've got our mask. Now go to run. My IDW got placed underneath my coastal zone file, so I'm going to turn that coastal zone file off. What this does is this is interpolating salinity. There's no extrapolation with IDW. Remember, it's interpolating salinity based on minimum salinity and maximum salinity that was observed at those points. It looks pretty good. Does everybody's look kind of like this?
Mine did the opposite. Mine made all the background the colors, and there's the still one color in the center.
You got something switched. You got
Awesome.
something
Amazing.
So that we're gonna
Beautiful.
do this again with spline and kriging. So I would just you have a second to redo it. Go to that IDW slide, Lauren, and try to rerun it. It's something that happened in environments, I would assume. So delete the current IDW in your art catalog, remove it from your table of contents, and then just slow it down a little bit, and just you probably just got something switched with like processing extent. It can happen like if you have a rolly mouse and you're over here in environments like processing extent, like if we go as specified below, you were like intersection, current display, you can do all kinds of wacky stuff, like just not even needing to. Okay. So one thing about raster data is the way that you display your data matters. So when you're looking at so we're looking at average salinity. This average salinity corresponds with these marsh types, right? And so marsh types, like anything under point oh, I don't know exactly what it is because it's like something, anything under like 0.1 is considered fresh. Anything under anything under like 1, or I think it's anything under 3 parts per thousand is considered intermediate. Anything between 3 parts per thousand and like 15 parts per thousand is brackish, and anything above 15 parts per thousand is saline. And so what the symbology has done is it's just kind of like arbitrarily selecting some divisions, right? When you are working with rasters and when you're working with things that involve a closer look at like classification methods, you need to pay closer attention to the symbology. So we're going to hop over here to symbology. And look, it's got primary symbology, it's got classify, okay? You can go and you can select unique values. You can go and you can select stretch. And stretch works pretty nice too. Under classify, I actually don't mind it being classified. This makes sense. But what we can do is we can change the number of classes. We could give it something like 20 classes. And so we could make, because really what I'm looking at, like if it only has 9 classes, there's just this huge area up here that's just like, below 0.2, this is not separating intermediate marsh and fresh marsh very well. So I want more classes, first of all, and second of all, I want more specificity at the lower range of salinity. And so what you can actually do is you can get particular, and you can change this, right? So like here, if you go over to histogram, right, it's showing us these class divisions, right? I've got everything between 21 and 16. I could go in and I could, it's kind of hard to manually do this, but you could scooch these class divisions up. I mean, you can like really, really, really get fussy about it. You can freeze your computer screen and make it go black if you're lucky. What I would really like is if you could fool around with the upper value a little bit easier here. And so like the upper value would be like everything fresh, let's say like 0.1. Let's see if she'll listen. She will. So now notice it's switching to manual interval. I can go here. God, I don't like it. Blah, blah, blah. And I could do 0 point. So everything under that's fresh. Everything under 3 is intermediate. Right. And then, and
And just to confirm, this is using the data of your coastal zones, right? This has nothing to do with the CRIMS data.
no, this is everything to do with your CRIMS data.
So this is okay, I guess I'm just kind of confused on how it's correlating because I'm trying to like think of how I would use it with my story map data, if that makes sense.
So this is taking these values, these average salinity values
Yeah.
that are at these points at your CRIMS locations that were recorded by your CRIMS locations, and it is predicting across unsampled areas. So it's just the same thing as like you take an observation and based on this collection of observations, the model is trying to understand, and trying to predict. So like we have an observation here, but I want to know what salinity is out here, but I don't have an observation. So the model is just predicting based on that IDW equation what the salinity is going to be right here.
Okay, awesome. Thanks.
Yeah. And so I'm fooling around with boop-a-doop-a-doop, the symbology over here. I'm not going to spend too much. The symbology for, here, I'll say this. We will fool around with this more in a little bit. The symbology working with interpolations takes longer than performing the interpolation itself. This is the art part of this, right? Getting your symbology right really, really matters. We're not going to use the IDW too much more, so we're going to stop because it takes forever. But we will fool around with this when we get to kriging and spline. So let's go back over to our geoprocessing tab. And actually what first, I'm going to make I'm going to go to natural breaks. I'm just going to leave it like that. Natural breaks 20. And now I'm going to hop back over. I'm going to save this before it completely shuts down and I can't do anything else. Okay. Now we're going to go back to geoprocessing, right? And so you might just have to back out of this IDW environments a little bit. And we're going to go back into this interpolation tab. And now what we're going to do is we are going to do an interpolation using a different method. We're going to use kriging. So remember, kriging lets you extrapolate. Kriging really, really wants to create a smooth surface, but kriging doesn't have to pass through the exact observations. So even though this particular CRIMS station right here, the average salinity was recorded as 4.76, kriging might be like, man, I think to make this model smoother, I'm not going to go exactly at 4.76, right? So that's what kriging does. So this is good follow-up to Taylor's question just a second ago. The input point features, I'm creating a raster from a vector data set. This is the vector data set. It's the CRIMS Hydro 2020. I am asking it to interpolate salinity, average salinity across this surface. I am naming this Krieg 1. We're going to leave all these semi-variogram properties the same. I am making my output cell size 500. I'm hopping over to environments and I am saying my output coordinate system is the same as my map. I am saying that my processing extent is coastal zone. I am saying that my mask is also coastal zone. And then I'm running it. Did you figure out what you did wrong, Lauren? No, we'll run this one. Let's see how she looks. Do you have something selected on your map?
I did before, and then I unselected it, and I did it again, and it still did it opposite.
Do this one, and if it doesn't work right, then you're going to share your screen, and we're going to get you sorted out.
Okie dokie.
And so I'm running kriging. We'll see how she looks. So this is lots of really complex math happening here in the background that we're not interested in. We're just happy it's working. So you should get something that looks like this. What do we think? Does it look like this? Your range should be 0.047 to 16.563. Sometimes it's a little bit different, but we're all using the exact same data set. Should be the same.
I'm gonna share my screen.
Okay.
But I had to join from my other computer, so it may take me a second.
Okay. Everybody else doing okay? There might be something corrupted with your original CRIMS data file, unfortunately. If it's running incorrectly every time, then it could be something with that original file. So that's going to be the first thing that I look at, is that original CRIMS file. That should be the first thing that you look at too. Make sure she's got a PCS, make sure she's got 380, 400 points, something like that. Because this is all based on how that original CRIMS file looks. Okay, turn off your basins. Let me see Krieging. Krieging completed with warnings. It looks right. It looks identical to mine. It's identical. It's totally fine. Was it just under coastal basins? Yeah. Good news. It's fine. Glad to help. Glad to help. Okay. Oh, I love this. I love everything about it. It happens all the time. It really does. I'm just like, it's gone. It's totally gone. It was just under your basins file. All right. So now we're going to do spline. So you're going to go back on geoprocessing. And we're going to go back to interpolation, and we're going to run, oh, you can't see anything yet. Boop-a-doop-a-doop, there. Back to geoprocessing, back to interpolation, and we're going to go to spline. And we are going to do the same thing. CRIMS, average, name it, spline 1, save, output cell size 500, environments, output coordinate system, extent is coastal zone, mask is coastal zone, run. Okay. So the thing about spline passes through assigned values, right? So at every single point that we see on this map, the interpolation adheres to whatever the value that we told what the value of average salinity is. So that's one good thing. But one thing that spline does is go over here and look at your classes. Because it extrapolates, it's giving us a negative salinity, and it's giving us a salinity of 50 parts per thousand. Neither of these exist, right? It doesn't know any better. It's a computer program that is doing its best. It's like, oh, well, it was pretty salty. out here, so I'm just going to assume that it's getting saltier and saltier, you know, times infinity. So it is doing its best, but its best means that it far, far exceeds the range of actual observed values. And so what you can do when you have a tool that we like in this situation, like Spline, and I'm going to tell you why we like Spline. We like Spline here because Spline respects the observational values. And we know that those observational values at crim stations are truthful. We have a lot of confidence in them, right? So we want to use Spline because we like what it does with those observed values, but it's giving us this crazy range of data. So what we need to do is we need to go in and we need to fix this. And so now we're going to use, if you all haven't done any coding yet in any of your classes, here's your first chance, right? This is this very simple, very, very short line of code. And so where you'll find this, so if y'all don't have your PowerPoint open, open it up because I'm going to make you copy paste this because it gets so much easier when you copy paste. Where it says name of spline, it is the name of whatever your raster file is that we just made. Mine is spline 1. It says name of spline. That's just whatever your spline is named. Let me show you. So what we're going to do is we're going to go back to our geoprocessing tab. And under Spatial Analyst Tools, you're going to expand this guy called Math Algebra. So there's only one tool available in Math Algebra. It's Raster Calculator. So expand that, open her up. And what we're going to do is we are going to copy paste the expression, this guy right here, the whole thing, copy it, paste it in this box. Where it says name of spline, that has to exactly be the name of whatever your spline is in both places. Code is exact. Every space, every parentheses, every quotation mark, every underscore, every single thing has to be precise. If something is going wrong with your code, 9 times out of 10, it's literally just you've made an error in the way that you've written it. Like for all of us, it just drives us absolutely bananas. So what this is doing, I'm adding something called a conditional statement. And so this conditional statement says that if spline 1 gives you a reading that is below 0, just interpret that as 0, right? And then we have to add in this other spline 1 code, okay? Our output, it's going to make a new raster with this map algebra on it. And so we're going to create a new raster. And so this is going to be spline. What do I say? What do I say I want its name? Okay, like tell y'all what I want you to name it. Spline. I don't care. Apparently I don't care what the spline is. Let's just do spline 2. Okay, and then go to run. And it's going to create a new raster data set that does not go negative. And now we have this nice stretched data set. Maybe it's not nice, right? Nice, not nice. And we, for salinity, for something like elevation, stretched data sets are really, really nice. For salinity, I like classes a little bit better. So we will fool around with symbology. Everybody got it? Everybody here? Cool. So with spline selected, I'm gonna turn all these guys underneath off. And I'm gonna just minimize them a little bit. I can also add a conditional statement that tells and so that's actually something that I should do, but in the interest of time, we're not going to do that. But I would add the same conditional statement Actually, maybe I'll go ahead and do it. If you guys want to do this with me, I recommend it. You've got your geoprocessing tab open. I'm going to take this spline that I just made, spline 2, and I'm going to say if it's greater than, what was my max salinity? My max average salinity was like 22. I'm just going to say if it's greater than 25, that's going to be 25. And then I'm changing that name to spline 2, spline 2. And then spline, I'm going to rename this spline 3.
You don't have to mess with environments.
Because we've already done it. We already have this base. It's all this is doing is it's a conditional statement. And so a conditional statement takes a data set like our raster that we made. We're not interpolating anymore. There's no more interpolation necessary here. We're just taking that raster data set that was already interpolated, and we're just applying some pretty sparse conditional statements. We're just saying, listen, if it's below 0, it's 0. That was what we did first. And then we're saying, listen, if it's above 25, it's 25, right? So just adding some guardrails essentially on
Okay.
on what we're doing. Sorry. Oh yeah. Stephen, Nicholas, you guys are the same thing. Okay. Yeah, no, nothing like that. And so I'm gonna try this. Actually, don't know if this code runs. We're gonna find out. And then we should have something that looks identical to this, except the range of values is in between 0 and 25. Oh, yeah, I didn't like that. Okay, so it was something with the way that I entered my code. Let me see if I can figure out why. Oh, no, I have it. Oh, look, I have the code in there. Native spline greater than 47. Oh, okay. Greater than 47. I say 25. 25. Oh, I'm missing. What am I missing? Okay, so I didn't realize that I even had this written down. This is what I'm telling y'all to do. I'm just changing this to, instead of 47, I'm changing it to 25. And let's see if I can figure out what I did wrong. 25.0. Comma. 25 minus 2. I'm missing space. Spaces don't always.
It was just the point oh. I just did it. It
Was it
worked.
just the point out?
Yeah.
It's so fussy. Yeah, space. That's a good. That was a good try, Alexis. And sometimes that like spaces do matter. Encoding spaces usually don't matter. It's just kind of for our brains. I'm gonna try to run it.
I have a
Oh,
point oh on
Mine's
the
saying
first
the
one.
map for
it's because it's supposed to be spline.
working.
Spline 2. Output raster is that's what. It's because it won't do it and rename spline 2. I got to do it spline 3. There we go. That should work. Oh.
I did 25.0, and then the one in between the commas, I just have 25, no point oh.
Where's that? Wait a second. Let me try my. Oh God. Conditional statement. Spline 2. If you are greater than 5.0, then you are 25.0. That is the way that I have the code written. So it's almost like I've done this before. Spline 2. I'm just trying to be like different and do it with a different value because I think that value makes more sense. Spline 3. Okay, let's try this one. my spline 2 is gone now. Hang on a second, everybody. Ooh, yeah, my spline 2 got messed up. Okay, so I actually have to remake all my stuff. Hang on a second, just delete that. It's not a big deal though, it doesn't take very long. Geoprocessing. Spline 2. Redoing that I'm redoing that first conditional statement. B. Delete. Paste. I think what happened was I screwed up the first line of code and I named it spline 2. And so, wait. And then it like corrupted that original file. Okay. I'm starting to like, I'm looking at the word spline and every time I look at it, it looks more insane. Do you know what I'm like? I was like, what's wrong? Like, why does it look so crazy? Okay, and so now conditional statement, spline 2, where you are greater than 25.0, you equal 25, you are named spline 3. Yeah, okay. Okay, so now I have this file that has realistic, reasonable salinity ranges. And that didn't, I mean, the coding stuff takes a little bit of time to figure out, but what we really want to fool around with here is the symbology. So it has automatically switched it to something called stretch. I don't really mind. I don't know. It depends on if you want unique classifications or if you want to keep it stretched. If you want to keep it stretched, I don't really mind that, but this black and white, not super helpful. I like this kind of unicorn rainbow color scheme that gives me a lot of variability. Yeah, it's something with your code, guaranteed. Or you have to do the same thing that I did, Nicholas, where you have to like recreate your spline because you didn't change a name and you have to rename it or something. It's definitely something in your code. Okay, but don't worry about it too much because this spline is actually meaningless. And I'll tell you why. Because salinity and the way that water moves across a human manipulated landscape is different. So we have all of these things across the coast that are going to act as barriers to salinity. Yeah, you do copy the thing from PowerPoint. Here. We'll mess with it when we get to it on the next one so that we can move forward. I promise I'll get you sorted out, Nicholas, if it still doesn't work next time. Okay. So now what we're going to do is we're going to run a new interpolation and we are going to actually run something called spline with barriers where we incorporate levies into this. So we have a levy file. Go over to your ARC catalog and you're going to expand this data set for lab 7. Oh, did I not unzip that file yet? I don't know if y'all did, but I apparently didn't. I'm going to go in here and go into levies. And so it's a zipped file with 2 zipped folders in it. So we've got to unzip these. Refresh. Okay. And expand both of these folders. We've got levied area and we've got system. One of them gives you polygons. One of them gives you polylines. I'm not sure why they've organized it this way. This is something that I got from like, I don't remember who, but they're fairly up to date. And so we're going to use polylines instead of polygons. Polygons should work fine for this type of analysis, but polygons, since we have it, let's do polylines. So you're going to pull this polyline in, and it gives us have parish levies in here, so we're still going to be having some uncertainty, but we've got state and federal levies. If you expand your attribute table, you can see the information that's associated with that. We've got address, we've got the name, we've got waterway, we've got, oh, we do have a couple parish. We've got, oh, Louisiana State, Angola's levy, right? Lots and lots of stuff. So Nicholas, are you doing the AME?
Yes, ma'am.
This might be a helpful file for you. There might be something about the AME here that you should look at. So remember this file and just keep track of it. But this one, of course, is not in the right projection, and we don't want to corrupt our original file. So let's do what we do best and export this data set. We're going to call this levies. We are going to give it a projection. We are going to run it. Okay. And then I'm going to remove the original polyline data set from my table of contents just to make everything as clear as humanly possible. Okay, so now what we're gonna do is we're gonna run the spline with barriers tool. All right, so go back over here to your geoprocessing tab, back it out, hop back up here to interpolation and look at this little friend, spline with barriers. Open that up. Just like before, input point features, CRIMS Hydro 2020. Just like before, our interpolated value is average salinity. Our output raster, this is where names start to get complicated. We're going to get fussed at some point if we have a really long name. I'm going to name this SPL, and I'm just going to go with barriers. Just WB with barriers, okay? Save that. Output cell size 500. Input barrier features. Levies. Simple as that. As long as everything's in the same projection, we're ready to rock. It's not the same projection, it's garbage. Everything's in the same projection. Our analysis is going to be right on. Everything looks good. So for whatever reason, environments, the mask doesn't work for spline with barriers. It will give you the most insane raster data set. Like you can run it if you want to run it and see what I'm talking about, but it gives us like a very silly looking raster data set. And this is just how ArcGIS is sometimes. It just gives you crazy stuff. It just has trouble. So what we're going to do, our output coordinate system is the same. It's the same as our current map. Our processing extent is the same. It's our coastal zone. We are not going to touch this mask. All right. We're just going to go to run it, see what we get. My kids just got home. Okay. So you see what we get when we don't set that mask, right? It's basically going, it's, here's my pixel. It's going 500 kilometers or 500 kilometers away from this point. We get this great big old rectangle. All right. We also still get negative numbers. We also get, the positive numbers are a heck of a lot more reasonable though. And we have clear distinctions in areas where we have levied stuff, right? So this is actually just showing the spline from earlier, right? It is not interpolating salinity in these barriers, right? It's like this is a town, this is a area, like, right, I'm not interpolating salinity across these barriers. It makes a lot more sense in the real world ecologically, but it looks like trash, right? It's this big rectangle. I don't want to use this. I don't want to look at this. Our workaround for this is to hop back over to geoprocessing, go into the extraction tab under spatial analyst, and you're going to go extract by mass. Our input raster is this spline with barriers. Our mass is our coastal zone. Our output raster is going to be SPL, WB, mask. Extraction area inside, go to run. So this is just like making a cookie, right? It's just a cookie cutter where it just takes the outline of the coastal zone and just stamps it out, right? So nothing with environments, nothing with anything like that. It's just going to give us a spline that looks like what we want. And so I'm going to turn this other spline off. Okay. So now I have the same problem that I had earlier where I have salinity that goes negative, which is not what I want. So I'm going back to this conditional statement. All right, so go back out of geoprocessing. Scroll down to raster calculator. Copy that conditional statement.
Did we do inside or outside for the extraction area? I'm sorry.
Inside.
Inside?
Mm-hmm. Yeah,
And did we change the extent at all?
Nope.
Okay. Thank you.
Yeah. Okay. So remember, everything that I have that says that's in red, this is just an example. You have to change this to whatever the name of this spline is. Mine is SPL underscore WB underscore mask. So SPL underscore WB underscore MSK. And you can just highlight it and then go up to the folders under algebra expressions and double click. But for short names like this is what we're doing. So again, same name, SPL underscore WB underscore MSK. Output raster. Spline. I'm going to name this SPL WB con. That means I have that conditional statement with it. Save. Run. Nicholas, let me know how this goes for you.
So we don't need the clipped with barriers in this one?
It's whatever your spline is named, whatever the name of your spline goes inside those quotations is the name of your spline. So whatever your spline is named, mine was SPL WB MSK. Yours is probably something similar. Everybody about here?
No, it's not working.
Okay, share your screen.
So this is the code, right? I follow all other steps. I don't have the other three splines, but from the other stuff, have I got the same file you did? Now, I don't know what I did wrong from there.
Turn off. Wait, so you have spline with barriers, mask. Okay, and that gives us our negative. Okay. Conditional statement. I can't tell either. I just can't see any. I'm like inside my computer trying to look at it. You've got conditional, spline WB mask, MSK.
The thing that says help is the with barrier.
No, that's fine because the black and white file looks fine. Open up your under output raster, open up your folder under out to the right. Yeah. Yeah. Let's see where you're putting everything. Okay. So you all, no, SPLWZ. Okay. Close it out. Go back, just like back out. Go over to your table of contents, turn all your other stuff off except for that spline WB mask. Okay, yeah, that's good. Okay, do me a favor, remove that SPL WB mask from your table of contents, and then I want you to re-add it from your catalog. Yep. Okay, now go back to geoprocessing, toolboxes. SPL underscore WB underscore MSK. Same right there. Okay. Output, no, you gotta name your output raster. And name it like, yeah, CON. Okay, try that. Right. Okay, let me see that exclamation point.
Say again, I'm sorry.
That says fix the missing parameter down at the bottom. Expand that.
It's not letting me.
What are you missing? Is something I can't like read and see if something's like misspelled.
It has an X. Oh, here we go. Let's see. File string line one con raster.
Oh. Oh. Go back to your output raster. Delete that name. No, just get out of there. Just get out of there. Just go back to like, yeah, and delete that and just do like, go ahead and just expand the folder again. Okay. Yeah, and is it where you want it? Yeah, put it where you want it. And just name this something like SPL underscore A. Okay, now go to save. Now try to run it.
No, nothing.
Okay, it's something with it's like not understanding where your original file is. I just can't see anything. It's really named SPL WB underscore MSK, right? We're like positive that's the name. No. 0. parentheses. Master files missing or invalid parameters. Delete the whole thing. The code, the whole code. And instead of copy pasting, try typing it in exactly. CON. Here, I'll tell you what to do. CON parentheses, parentheses, quotations, name of your spline, which is SPL underscore WB underscore MSK, quotations, less than symbol. Did that one not come over? That might have been, and comma, parentheses, comma, parentheses, good. 0, comma, double quotations, and then the name of your spline again. Quotations, parentheses, run.
I think he typed it wrong.
Huh?
Like, yeah, he put comma. Yeah.
Yeah, yeah, yeah. There we go. There we go. Okay, now it's right. So when in doubt, don't copy paste. Ooh. What's going on up there? Okay. All right, so you're set. That's right. That was frustrating. I'm sorry about that. I don't know. I
It's
wasn't.
okay. No, sorry.
Was it the greater?
I was about to.
Did that just not copy paste properly?
Um.
Hmm.
I can.
No, we don't. It's fixed. We're gonna go ahead and stop sharing, and then I'm gonna and we'll see what we can get after. Okay. Okay. Share again. Okay, I'm glad we got it sorted. All right, so now we have this spline. It looks good. Okay, let's fix our symbology so that it's not just black and white. Again, I really like this unicorn color situation that goes all the way to pink. Where is my girl at? One I love that one. It's stretched. If I wanted to have more division up in these fresher areas, I might do something like unique values, classify. You can do all kinds of different things there and play around with it. I encourage you to play around heartily with your symbology. So if we go up here to explore on your ribbon, and you're going to expand this, we're going to, where's the identify tool? Oh, hang on a second. Where's the identify tool? I do this every time because this is one thing that's really different. Yeah, just click somewhere on your map. Where are you? Here we go. And it's going to pull up this pop-up that's going to give us the value wherever we clicked at the top that's the topmost layer. And so what I actually want, nope, it's not that. I want to identify, where's the identify tool? Explore, navigate. Hang on, I'm trying to, I do this every time where I just like zone out and forget how I do this. Navigate. Oh, so annoying. There's like just a simple like thing that you change where when you click on it, it will give you the value for all of the layers that are on your map. Navigator, camera, magnifier, explore. Really thought it was navigate. Not you? oh, I do this every semester that I've been teaching this, and I think that I wrote it down, but I didn't write it down appropriately. And it drives me crazy. Okay. Well, I'm gonna figure out how to do this another time when it's not 5 o'clock. Okay. So we get, depending on like which spline method, which interpolation method that we used, we're gonna get sometimes wildly different answers for what salinity is. So understanding which interpolation method to use, super important. You have to know a little bit about that. Okay. So remember a million years ago when we downloaded that master plan coastal vegetation data? Let's look at it. So hopefully you've already extracted it. Hopefully it's all running. What we have here are a suite of different modeling scenarios that show how vegetation type changes given if it was like a maximum sea level rise scenario, if it was a minimum sea level rise scenario, if the Mississippi River lost all of its water, if there was no sediment. These are different scenarios. See this S00, see this SO1, SO3, SO4. These are different modeling scenarios based on the range of possible futures that we have. You can figure out exactly what these are by looking at, oops, nope.
Oh, I'm sorry.
By looking at the metadata, right? And so we have this nice metadata folder. It's going to open up a web page. That web page is going to give us all sorts of good information on why these data were collected, what they mean, what the scenario runs are, right? And so here it's like, this is what this one is under environmental scenario without the map. So, whoa, where'd she go? Essentially, the first one, this is coastal vegetation under initial conditions. So this is like the base of like where everything starts, looking at coastal vegetation. Coastal vegetation under low environmental scenario without the master plan. So the master plan basically is like, we have all of these potential restoration projects and we want to see if sea level rises a half a meter, do any of them do anything? Do any of them provide any benefit towards ecosystems and communities at all? If we do no restoration projects and sea level only rises, 0.25 meters, doesn't matter, right? And so basically this is this suite of possible futures that we're looking at to try to basically identify the collection of restoration projects that have the greatest benefit. Benefit to people, benefit to habitats, benefit to species, right? All of those things together. And you can figure out a lot of details about this by looking at the metadata and then by digging in a little bit deeper into the information that's available online, right? But so what we're going to do is we're just going to pull this initial conditions raster data set over. So grab her and bring her on over. I expand it. Not really. Put it right above my spline. Okay. So this is why metadata are important, because when you get a raster data set, it's just going to give you a value. And that value could be elevation, that value could be some categorical, assessment. In this situation, we're getting these values 1 through 8. And the only way that we can know what the hell that means is if we went over to our metadata and we looked at what it meant. And so we've got 1 through 8. 1 is forested wetland or swamp. 2 is fresh. 3 intermediate. 4 brackish. 5 is salt. 6 they don't have, but 6 is water. 7 is bare ground. 8 is floating marsh. So now we have a little bit of context for what we're looking at. You can zoom way in and you can see how it's pixelated, and you can see where we have differences, right? So this was forested, this was fresh, this is intermediate, this was brackish, this is water, right? You know, we can go in and we can see what all of those are supposed to be, and we can change the way that these are represented so that they have more meaningful, you know, colors, and we can change the labels and stuff like that. We'll do that in a few minutes. But what we're going to do is we're going to do something called reclassifying. So that we just get everything that's land and everything that's water. And so for some of y'all that are working on forested habitats, so there's a couple of y'all in there that are interested in longleaf pines. And there's a couple of y'all that are in here, you know, that are looking at, you know, comparison of habitat types across time or something like that. Reclassifying rasters can be immensely helpful just from a data management standpoint, because you're like, I don't care if it is early successional abandoned agricultural field or if it's an active agricultural field. It's agriculture, it goes in this section, right? I don't care if it is a, you know, managed pine forest or a non-managed pine forest, right? Like maybe there are meaningful classifications that you might want to make. And so what we're going to do with this data set is we're going to make some of those meaningful classifications. So hop back over to geoprocessing, back it out until you're in the in the spatial analyst tool. And you're going to go down to reclass. And under reclass, you're going to select reclassify. Our input raster is this big guy. Our reclass field, notice that we only have, you can either do the number of pixels, the count, or you can do the value, right? We're going to do value. And so what this is value 1. You have to know what the metadata are, right? And so I'm going to pull this over to my other screen. You all should do the same, or you can follow along with me. But 1 is swamp. So that's land. 2 is fresh marsh. So that's also land. So I'm going to give that a 1. 3 is intermediate marsh. That's also land. 4, brackish marsh. Again, land. 5, saline marsh. Also land. 6, I have hit not land. I've hit open water. Okay. So I'm going to give this guy 0. All right. Bare ground, that's land. Floating marsh, is floating marsh land or is it water? It depends on who you talk to. I'm going to say it's water here. Last time I did this assignment, I said it was land, right? Floating marsh is funky. It's crazy. So basically what we're doing is we're reclassifying this raster. We're saying there are only 2 separations. You got a bunch of land, you got a bunch of water. All right. I just want, and this can be really helpful for creating background data sets that don't give you that yucky looking Esri situation. And our output raster, I'm going to name this land. Actually, I'm just going to name this L_W, land and water. I'm going to go to save, and I'm going to go to run, right? Right? Yep. Go to run. And this is going to create a new raster that just has 2 values, some 1s and some 0s. And an 8. Oh, because I didn't change 8. Damn it. Okay. Did you guys change it?
When you go to change it, it just automatically changes back.
Automatically does it. Okay, so what we're gonna have to do, and sometimes that happens. So what I'm gonna do is I'm gonna put this back in.
That happened for me, but I was able to force it, and so it became a 0 now for me.
Uh-huh. Yeah, you can force it, or you can just remove it. Here, I'm just re-adding it. Okay. I'm gonna go here. I'm gonna go to symbology here. And I'm just gonna go to symbology and I'm just gonna select 8. And I can remove that. Okay. I can just remove that, or you can force it to read it as 0. You can reclassify it again. There's lots of different ways that we could do this. And so my point in doing this and reclassifying this raster is because I want to make a water mass. And so I'm going to go back over to geoprocessing. I just want to see if I can do it just because it pisses me off. Just want to know. Still wants that 8. Okay. I don't always do this. I'm not sure why. Add it to the list of things that we don't entirely understand why it does. Okay, but what I want is I want a mask that is just water. So we're going to cruise up here back into Spatial Analyst Tools, and we're going to go to Extract by Attributes. And let's open up this Extract by Attributes. My input raster is going to be this land water file. And I'm going to extract where the value equals 0. Okay. So I'm pulling out everything that's water. And I'm going to name this water.
Wait, Dr. Demarco. Whenever did you just delete the 8 or did you?
I just removed it from the label, but it doesn't matter because we don't need that for right here. We're just pulling everything out. Yeah, it's floating marsh again, just giving everybody a hard time. Like, you know what? I wonder. I usually include floating marsh as land, and so for whatever reason, this problem doesn't happen when you do it that way. But it doesn't matter because right here, it just screens all of that out. So it's a good question. Sorry, Lee. All right, go to save. Output raster is water only, where value is equal to 0 for our land water raster. Run. Rasters always take a little bit longer. Okay. And it's giving me, for some reason, this weird stretched value, which is okay because all I have to do there is just change. Ooh, this looks trippy. All we have to do is change the symbology so that it's just unique values. Yeah. Just everything. There's only one value. So everything that's a 0, we can make a really deep blue. Off. Okay. Doesn't that look pretty? Pretty. So now my 2 rasters that I have visible on my map right now are this spline with barriers, my beautiful unicorn spline, and this land water mask. So crims, like really what we're trying to do, you know, so you might be asked the question is like, if are you trying to interpret salinity across a marsh surface? Is it accurate to interpret salinity across land? Or should we just be interpreting salinity across water? Maybe you have a species that is just important in the water. Maybe you have something that's just on land, right? This is important because a lot of times you're like, I don't want to show, you know, I want to pull water out, I want to pull land out, I want to pull something out based on size or based on location or based on attribute. You want to extract by some reason. And so what we're going to do here is we're going to extract, so we can see the salinity in land, right? What we're going to do is we're going to do one more extraction, and we're going to extract by mask. So go back over to geoprocessing, extract by mask. Your input raster is your spline with that, the last spline you made, good spline, with barriers, with that conditional statement that worked. We're going to extract it based on that water file that we just made. And we're going to name this spline, SPL, water. Extraction area is inside. We're going to go to run. And then we're going to have an estimate of salinity that is more accurate than the previous one, potentially, because it's just in the water. All right. We can fool around with symbology, make sure, you know, you can stretch it, you could classify your values. A lot of this magic happens again in the background, just with symbology and the art part, right? Yeah. So you can see where it gets nice and salty. You can see where the Mississippi River pours out, super duper fresh, starting to look pretty cool, pretty cool. Let's save our maps. Okay. So let's do something else really quick. Yeah, I guess. It's just this lab lasts so long, and like I know everybody's about to die. But this last little bit working on focal statistics for this lab. This is a cool way that you can calculate edge, right? And edge really, really, really matters for a lot of species. It really, really matters for a lot of different types of habitat management. And so calculating edge means calculating focal statistics. And so running your focal statistics in on a raster data set. And so basically what we have is we have this land water file, this land water data set. And maybe instead of having something that's quite this clear cut, we want like relative landiness and relative wateriness. And so what you do here is you run focal statistics using nearest neighborhood statistics. So we've got a pixel and we've got all of its neighbors around it. And so if your pixel right in the middle of your zone is all land, but everybody to the north is water and everybody to the south is land, then that pixel is going to and not, instead of being a 0 or a 1, it's going to create like a range, right? Where as it's closer to 1, it's more landy. As it's closer to 0, it's more watery. Thinking about this from a terrestrial perspective, you could have interior habitat and edge habitat, right? And so you would look at some, you know, the whatever. And so when you reclassify your raster, you just have to remember whatever that value is that you assigned to your interior habitat, and whatever that value that you assigned was to your edge or something, or your you know, your habitat transition, right? So let's say that you have a forest and you have a field, and everything that's a forest is a 1 and everything that's a field is a 0. Anything that was, you know, lower than 0.5 is more fieldy and more edgy. Anything that is below higher than 0.5 might be more interior. So what we're going to do here is we're going to go back out to geoprocessing, and we are going to expand this neighborhood tab. And under neighborhood, we have focal statistics. So open up focal statistics. And for our focal statistics, our input raster is going to be this land water data set. Our output raster, let's name this something like edge 1. Neighborhood is going to be rectangle. Statistics, it's got width, it's got height. It's basically like I have a pixel and it's like how many pixels above do you want to use, and then how many pixels to the side and how many pixels below, right? And so if you're think about the way that your squares work, like if y'all are in the computer lab, look down at the floor, right? You've got a pixel, you've got a square in the middle. Do you want 3 that go above? Do you want 6? Do you want 9? It has to be divisions of 3. So let's actually go up and we're going to change this width to 6. And we're going to change this height to 6. And statistics type, mean, oh God, you can do a lot of different things here. That's more advanced GIS. We'll stick with mean for now. And we're just going to go to OK. So as you can see, right, most of the time that it takes to make these maps is taken up. We haven't even like messed with symbology too much, but think about like to make a beautiful map that's precise and says all of the things that you need to say, you will have to fool around with symbology for a pretty long time. To get everything in the right place, you have to reclassify, you have to export all of your data. Oh, I have an 8 here still. You would have to export all of your data. It's 8 here. Oh, floating marsh, you do not complete me. This is just me trying to do something different. I've done this lab so many times and I never do it with floating marsh. Let me see if I can get out of here. No. Yeah, you did fail to display. Okay, so my edge 1 sucks. Okay, so what we have to do, we have to remove this edge 1. We have to remove this land water file. Carl, how did you force it to go to 0? What did you do? Do you remember?
Yeah, when we were doing the reclassify, when I noticed that it had the 8, I undid it so that it wasn't there, like the thing itself. And then I just went back to the 8, and every time it would say, if when I put it to 0 and it would change, I just go back and put it to 0 until it stayed, and eventually it just stayed there
You just
as
kept
a 0.
thing over and over again until you got the result you wanted. Oh my gosh. Yeah. Okay.
I mean, it only took like 2 or 3 tries for it.
Okay, that's good. Okay. Oh, that's Jacob. Yeah, Jacob kept doing the same thing. Okay, so what I'm gonna do, guys, stick with me. We're gonna reclassify again. We're gonna reclassify again. So go back out to geoprocessing and you're gonna go to reclassify. You're gonna open up that reclassify tool. Your input raster is gonna be your master plan scenario. Your reclass field is still gonna be 0. Everything is gonna be 1, including floating marsh. Floating marsh apparently was not pleased that it was not included as land. Just like floating marsh. And this is going to be land, water 3. I'm on land, water 3. I'm sitting here in like the darkness, losing my mind. Oh yeah, no, it does it again. Damn you. Yeah, you just have to like, that is so annoying. Mine will. Click the table? What do you mean, where?
Like click click on the table where it says 8 on like on the left.
Dr. DeMarco, instead of clicking out of it, I just hit enter and it listened.
Christ. On a crutch. Okay. There we go. Great. Yeah, I was just clicking in the wrong place. Clearly. I was just clicking. Oh God. This is like, I really need to break this lab up into 2 separate labs because it's so cool, but I'm dead. You guys are dead. We're all dead. Can you imagine staying here until, oh my gosh, destroyed. Okay. So this is running. It's going to be beautiful. I really want to do this classification because some of y'all that are looking at forests, you have access while this is running. Let me just take a second. I want to show y'all if I can find it. A couple of you all had comments from me about this data set. This is an incredible data set that shows land use, land cover for the United States over really, really long time periods, right? And so you can get change. This one. It's the multi-resolution land. It's this one. Just find her. I don't want to view. I don't want to view you. I actually want to see the data. Downloads. And so you can get herbaceous cover, shrub cover. You can focus on like specific areas. This is actually not the like method that I want to use. No. Oh God, they changed their website. So now it's just freaking viewer. View. Everybody loves these viewers. It's super annoying. Data are offered for download. I guess this is where we're at. Land cover change. 2023, 2021. You can go back and a lot of these, like if you look at these, let's see if it'll just let us look at the metadata. Land cover program, it gives you, it looks almost identical to what we're looking at, right? And so it gives us basically like this raster data set that instead of giving, our values are 1, 2, 3, 4, 5, 6, our values are 71, 81, 82. 81 is pasture and hay. 82 is cultivated crops. 90 is woody wetlands. 95 is emergent wetlands. And so let's say that you want to look at the amount of land that was covered by longleaf pine forests in one time period versus longleaf pine forests in another time period. You might want to reclassify that data set. You might want to go in and figure out what's land and what's water. Okay, good. So my land water looks great. You guys are all probably there. Let's go back to geoprocessing. We're going to go to focal statistics. Our input raster is this new land water file. Our output raster is edge to cell size. I'm going to do 9. I'm going to do 9. Let's do 9. 9, 9, run. This is going to be so worth it. So worth it. Yeah, after this wall, just after this wall, go stare at a lab for 30 minutes. It's brutal, but it's so cool. It really is so cool. We're almost done. So the reason that I decided to do this land water mask looking at coastal Louisiana is because this actually could be theoretically helpful. I know that increasingly we have lots of students that are interested in fish, and there are lots and lots of fish species that really, really like edge, right? There are, like, for example, redfish really like edge. And if you compare edge from 10 years ago to edge now, you'll see some significant changes in the area of edge, right? So now we have this raster. Pick an area and just zoom way in. Right, just zoom way in. You can turn everything off except for this topmost file, this edge file. And it looks kind of fuzzy, right? But that's okay, because then you can go into these kind of like back barrier habitats and you can check it out. You can click and you could just go, the value of this area is 0.09. So that's very watery, right? The value of this area is 0.54. That's a little bit more landy. And then what you could further go on and do is you could reclassify, and what we will do, we will reclassify this habitat to create a map of preferred or suitable habitat. Oh, and so this is really where like the important stuff is, right? We've been talking about habitat suitability. We've been talking about interpolation. We've done all of these things. I'm starting to get kind of broken up and insane. But what we do here is we're going to take this land water map, this edge map. And if you have a species that has some range of preferred conditions, which you do. So Delana, if you're still out there, you are going to have a range of preferred salinities. Lauren, Lauren's cracking up in the background. You're gonna have your ash borers are gonna have some range of preferred temperatures. Feral hogs can do anything, right? But you know, a lot of our invasive species are really fussy about salinities or something like that. So in this case, what we are gonna do is we are gonna reclassify. We're gonna do one last thing. One last thing. We are going to go back to focal statistics. No, that's not true. We're going to go back to reclassify. We are going to pull in this edge file. We're going to reclassify it based on values. And we're going to go to unique. So go to unique. That's not actually what I wanted to do. That's not what I wanted to do. Cancel. Cancel. Get out. There's a lot of screaming going on upstairs. We're going to go to classify. Are we? Are we going to do anything? Oh, it's over here. There it is. Okay. So in this example, we're going to create 3 different habitat types. We're going to create suitable, unsuitable, and preferred. So we just need 3. So we're going to leave this at natural breaks. We're going to go 3 types. The upper value here, we're going to say everything below 0.25 is for our species. Let's say that we're looking at a kind of shrimp, right? Oh, wait. Everything below point, what do I say? Said everything below and everything above. Everything between is 1. Oh God, I'm so tired. You know what we're gonna do? We're gonna stop because we have nothing. This is so cool. I'm going to show you guys how to do it at the beginning of class next week. It'll take us 15 minutes and we won't be sitting in the dark being sad. We're going to cancel this. Please save this. Remember, this edge 2 is exactly what we want. All right. Save your project. Next week, we're going to do these last couple of slides. And it's going to be beautiful, and it's all going to come together, and it's all going to gel. You don't have anything to do for this lab yet. All you have to do is work on your story map draft. That's all. It's the only thing to do. It should be fine. Fine. Work on your story map draft, make it beautiful. We'll take a couple of minutes and look at this next week because again, it's super duper cool. It's very high level GIS, but it's dark and we've got to stop. All right. Does anybody have any questions? Are we gonna go stare at a lab or stare at a wall? Okay, great, great. Let's go have drinks and stare at things. Everybody have a good night. I will talk to y'all later. Good luck this weekend. Holler at me if you need me, okay?
Can I ask you a question about my project?
You can try your best, Francesca. I'm not sure
I'm
what
sorry.
No,
Do you
no.
want me to just email you at another time?
No. I just go ahead. Y'all take off if you need to. But yeah, what